And thanks to the organizing committee for inviting me to be here. Can I move? There's no feedback anywhere, so good. Can you folks hear me at the back? Okay, good. So today, as Jim said, I'm going to be talking about this 5-year grant that we received from USDA through the NEFA program to look at feed efficiency in swine. It's been a really exciting time for us. We're just now well into our 4th year of the 5-year grant. And it's been a particularly exciting grant to be a part of because of the array of disciplines that we've been able to bring to bear to study and to all focus concurrently on feed efficiency. So we have nutritionists, of course, physiologists, microbiologists, behaviorists. We have we're studying proteomics, transcriptomics. We have people involved in bioinformatics. Quite frankly, a number of disciplines that I'm doing well only to be able to pronounce them properly because I really don't understand them. But that's the joy of collaboration and being able to bring new approaches to trying to understand feed efficiency. What's the biological basis, and where can we go with this information to provide something valuable to the industry? So we have involvement from 5 different institutions in 4 different countries. In addition to Iowa State, Kansas State is heavily involved in the project, as well as INRA. And the joy of the INRA group from France is that they also have a select line of pigs selected on the basis of residual feed intake, which I'll explain in a minute. And they've been doing really parallel kinds of studies, independent of us, but communicating back and forth. So we have an opportunity to compare our results versus their results, and they've used pigs that have different background and have been selected slightly differently. And that's really provided a lot of value to the program. And then the Australians are involved in the project as well as the University of Saskatchewan. So it's really exciting to see all this go on. So our overall objective of the program is to increase nutrient utilization, feed efficiency, remembering that when the feds came out with the call for proposals, one of the things they were looking for was the reduction in animal agriculture's dependency or demand and utilization on feed grains. And so that's part of our objective then, is to strengthen the competitiveness of the pork industry. That's important to the industry, but also reduce its demand on grains and proteins. And that's through research and extension. And another part of the project that's really interesting is that by definition, no more than 2/3 of the budget can be assigned to either research, teaching, or extension. And in our case, 2/3 of the dollars are going to research and 1/3 is going to extension. We don't have a formal commitment to teaching, but there's just, as you can appreciate, a whack of graduate students at both the master's, PhD level, and postdoctoral fellows that are involved in this project. So the specific objectives, and I'm only going to focus in on really 2 of them, and in particular 1 of them. But the first objective was to quantify the performance of pigs selected for increased feed efficiency on one dietary regime, but when you put them on a different kind of a dietary regime. So these pigs have been selected for 8 generations, we're up to 10 generations now, selected on a corn soybean meal kind of diet, so a higher energy diet. But what happens, as you can appreciate, 3.5 years ago, price of corn was very, very high, and we were looking at a lot of byproducts which are higher in fiber, and we thought it appropriate to say, well, what happens if we take these pigs that have been selected uniformly and uniquely on feed efficiency and nothing else over 8 generations on a higher energy diet and put them on a low energy diet? What happens to those pigs? And what happens to their supposed superiority? And so I'm going to talk a fair bit about that, and as well as the second objective, and that was to say, well, what about these pigs that have been selected for feed efficiency? How do they stand up to stressors, immunological stresses, behavioral stresses? I think we hear a lot, at least anecdotally, people saying, well, you know, these pigs are so high-strung, or they're more susceptible to disease, or whatever. We wanted to ask that question, and this grant gave us the opportunity to do that. Objective number 3 is really all to do with genetics. And in the genetics component, They're looking at trying to understand the genetic basis of feed efficiency, carry that forward, to develop tools that can be used by the industry to select for feed efficiency more effectively and more rapidly. And most critically, they're going to be taking that information and working with 2 breeding companies to apply the knowledge under commercial conditions to see if the tools, the selection tools that have been developed, in fact, do work under commercial conditions. And then objectives 4 and 5 really deal with extension, and I'll talk about those very briefly at the end of the talk. By the way, Jim, I compliment you as the chairman for selecting a room with not 1, not 2, but 3 clocks to make sure I stay on time. And I want to really compliment the audience because you guys actually stayed around. I couldn't believe it. I thought the room would be 3/4 empty. So I'm, the pressure's on, or you're very gracious. But it meant,
there's a door prize at the end.
There's a door. Oh, Wayne, you just burst my bubble, man. Is it scotch? Hell, I'll stay around for that one. So those are the objectives. And this is, as you can appreciate, working with so many groups and trying to explain this to reviewers and granting agencies, we had to break it down in terms of how the resources were going to be used. And this may not show up very well, but so by year we show the functions that are going on, and you can see we're right along here. So we finished things like the stress challenge and stress evaluation. We've looked at behavioral testing. We've looked at some of the nutritional components, but now the genetic and biomarker component is becoming much more active. We haven't really done the microbial analysis. Andrew Van Kessel, University of Saskatchewan, is going to do that. Great guy, and so that has yet to be done. And then the transcriptomic and proteomic analysis has yet to be done as well. Okay, so first off, what is RFI? What is residual feed intake? It's a different kind of expression of feed efficiency, and there's reasons for doing it, and it's really to try to average out, when you're selecting for feed efficiency, pigs that have different weight, different growth rates, and so on. That's how it originally started. Now residual feed intake is calculated on the basis of the growth of the pig and its back fat. And basically, what they do is they model out what the feed intake should be, or the energy intake should be, given this individual pig's growth rate and back fat, and then comparing that theoretical, what it should have been, to what it actually was. So if the pig consumes more feed than the model, then it's considered a less efficient pig. It has high residual feed intake because it's quote-unquote eating too much feed given its growth rate and carcass composition. And if a pig eats less feed than the model suggests, then that pig is said to be a low residual feed intake and is a more efficient pig because it's actually able to achieve that growth rate and carcass composition on less feed than projected. Does that make sense? Because that's going to be really important as I go forward and explain some of the research. So what's the impact of this after selecting over 8 generations, and remembering that the selection is for RFI and nothing else. This is not intended to be a commercial selection process. This is to look exclusively and uniquely at residual feed intake. And so after 8 generations, residual feed intake has declined by 241 grams per day, pretty significant. Feed intake dropped by 376 grams per day. Feed conversion dropped by 22 points. Back fat dropped by 2.5 millimeters. But average daily gain dropped by about 80 grams a day. Now, compared to the drop in growth rate, in feed intake, that's a pretty modest drop in growth rate. And we know that if you select exclusively on feed efficiency, you are going to be selecting against growth rate. However, under the RFI model, remembering when we're selecting where we bring in average daily gain and back fat content into the model, that should help to minimize that. And since the drop in growth rate was only 80 grams a day, I think we could conclude that it worked. And the loin eye area increased by 1.5 millimeters. not very much over 8 generations. So let's first talk about this, this question of the robustness of these pigs that have been selected for feed efficiency in terms of dealing with some of the troubles of life, we can call it, in the life of the pig. The first one was nutrition, and this relates to the first objective. And we, as I mentioned earlier, we said, what happens if we put these pigs that have been selected on a corn-soy diet and put them on a much more higher fibrous diet as well as a lower energy diet? So I think the point here is it's important, thinking back to Dr. Bach-Knudsen's talk, that we're talking about fiber as well as energy, right? And so we're really putting these pigs under pressure because we put 20% soy hulls, 20% wheat midds, and 7% corn bran into this diet. That's obviously that's a research model kind of a diet. That's not anything being fed commercially. So we increased neutral detergent fiber from 9 to 26%. We dropped net energy from 2.5 to 2.0, so energy concentration dropped by about 18%. And so we put these pigs under pressure. So what did we find? If we look at this graph, and we're struggling a little bit whether we show the average of these 3 generations or whether we break it down, and I think you'll see as you look at the next 4 or 5 slides, you prefer it, hopefully you prefer it that we show you the individual generation response. So generation 8, parity 2 pigs, generation 9, parity 2 pigs, and generation 10, parity 1 pigs. Okay, so that's who we were using. This is the first bar represents the low residual feed intake, and the third bar, low residual feed intake. I think that's red, but I'm colorblind and it's red-green deficiency, so it's whatever color you see, okay? But that bar and that bar is low residual feed intake, yellow, orange. That is the high residual feed intake pigs. The plain bars are the low fiber, so the high energy, low fiber diet, and then the crosshatch ones are the low energy, high fiber diets. So let's take a look, and we can see on our regular diet, indeed, we did see reduced growth rate in the low residual feed intake pigs in parity or in generation 8, in generation 9, although it was only a trend, and in generation 10. So we got that consistent response. However, when we went to the high fiber, lower energy diet, the difference is smaller, but we still see that the low residual feed intake pigs would grow slower than the high RFI pigs. So that was maintained. Now, looking at, that was feed intake, pardon me. I'm very, very sorry. So let me just go over that again. So the feed intake was lower in the low RFI pigs, as we would have expected on the control diet. And it was still lower, but the difference is a little bit less when we fed them the higher fiber diet. Now let's look at average daily gain. And in the generation 8 pigs on the control diet, we saw no difference. Generation 9, we saw no difference. In generation 10, we did see that the low residual feed intake pigs did grow a bit slower than the high RFI pigs. When we put them on the high fiber diet, basically continued to see the same trend, although everybody grew slower, as we would expect, because they certainly could not consume enough energy, or certainly as much energy as they were consuming before. So they were either, the two lines were either the same, or there was a greater intake in the high RFI pigs. My apologies for confusing you on that. Now let's look at feed efficiency. So we would expect that the low RFI pigs should be more efficient than the high RFI pigs, and indeed they were, when we fed them the corn-soy diet. When we fed them the high fiber, low energy diet, that advantage was lost. Now, this is a glass half full or half empty situation because we look at it and say, well, they lost their advantage. However, they weren't worse. So they didn't sort of crash on us and say, well, geez, we just can't handle this high fiber, low energy diet and become inferior to the high RFI pigs. They just lost their superiority. So obviously the conclusion is that when we're selecting pigs, we need to keep in mind the nutritional environment that they're going to be kept in as we move forward. And if as an industry if we are going to feed lower energy diets in the future, we're going to have to keep that in mind in our breeding programs. Let's take a look at back fat depth. And again, we expect the low RFI pigs to be leaner. Generation 8 they were. Generation 9, it's not significant, but numerically they're different. But it was maintained, the same trends were maintained even on the high fiber, low energy diets. And looking at loin muscle, we see not a lot of difference. We see some differences in generation 8, but remember it's a small difference that we're looking at anyhow. Okay? So they lost their superiority in terms of feed efficiency. They maintained their superiority in terms of back fat. So low RFI pigs fed the low energy, high fiber diet did not maintain their superiority in feed intake and feed efficiency. Pigs that are more efficient on one diet may not be more efficient on a different diet. But keeping in mind, this is an 18% drop in energy concentration. That's a kick, right? The performance under the LED, I'm going to show you this data in a minute on heritability. And we didn't have enough data to define genetic correlations. So let's take a look at some of the heritability data. And Dr. Jack Deckers is really the leader on the quantitative genetics part of this project. In fact, I should have said earlier on, my role in the research is pretty minimal. I'm a kind of a collaborator. I'm taking more of a lead role on the extension part of this project, and I collaborate on the nutritional side, but Nick Gabler takes a real lead on the nutritional part of the project. Steve and Elizabeth Lonergan on the meat science side, Chris Tuggle on the genomics side, etc. So I want to acknowledge they're all listed as authors on the paper and the manuscript. But let's take a look at heritability. And if we look at heritability on the high energy, low fiber diet, whether we're looking at average daily gain, back fat, loin muscle area, feed intake, residual feed intake, or feed efficiency, we can see that there's the heritability that we would expect, sort of in that 0.5 to 0.6 range, with the exception of loin muscle area. And indeed, the heritability is different on the two diets. So not only then is there the challenge of aligning our selection programs according to the nutritional regime that the pigs are going to be raised in commercially, but we're looking at differences in heritability depending on the diet that we're feeding.
Which changes your index.
Which can change your index, exactly. So this is something that needs to be kept in mind, because look at where the heritabilities here are 0.5 to 0.6. The heritabilities over here oftentimes are less than 0.2 or even 0.1. So that's fairly serious. Okay, so that's one part of what we looked at. Now, Dr. Rowland at Kansas State University has a really good, well, first off, he's got a really nice new facility. Secondly, he has a really good PERS model. So Dr. Deckers and Dr. Tuggle sent their pigs down to Dr. Rowland at K-State and exposed the low and high residual feed intake to the PERS virus to see how do these pigs respond. Do the pigs that have been selected for better feed efficiency, are they going to just crash when they're exposed to this virus? And so I'm just going to show one piece of the data, but it pretty much summarizes the conclusion. And this is the average daily gain of the low and high RFI pigs that were not challenged. And here's the average daily gain of those pigs that were challenged. And you certainly wouldn't conclude from that that the low residual feed intake pigs, the more efficient pigs, are less capable of dealing with an immunological stress such as PRRS than the high RFI pigs. And that's the overall conclusion that Dr. Rowland had. Now, whenever you embark on a research project, sometimes things happen that you don't plan on. And some of those things that you don't plan on aren't very good, and some of them turn out to be really, really cool. And in this particular case, when the pigs went down to Dr. Rowland, he called Jack and said, "Jack, what's wrong with these pigs?" Because they'd done some of their preliminary blood tests, and it turned out that some of these pigs were what were called skid pigs. They suffered from this phenomenon called severe combined immunodeficiency. Now, animals that have this immune deficiency, this has been found to occur, quote unquote, naturally in other species, like certainly humans. The bubble boy, if you may have seen that in the press, that's a skid human being. Horses, dogs, cattle, mice, but it's never been, to the best of our knowledge, it's never occurred naturally in pigs. It has been produced in pigs by knocking out genes, but it has not been occurring naturally. What this does is, this is totally serendipity, but it's opened up, Jack, after this happened, had the biggest smile on his face I ever saw, because this is a huge opportunity for his career and Dr. Rowland's and everybody else's, because they now have a model for studying the immune system. And this will allow them to use the pig as a model for everything from the effectiveness of vaccines, the development of vaccines, the study of cancer, etc. So this is kind of a big deal. The problem is you got to keep the pigs alive. And fortunately, we have Matthew Ellenberg in our group, or Ellsworth, sorry. He was familiar with doing bone marrow transplant in dogs, and so he thought, well, if I can do it in a dog, I can probably do it in a pig. And he had enough success that they're keeping those pigs going, so we now have skid pigs that can actually stay alive, but they're still skid pigs. So anyhow, that's just one of the side things that came out, and it's very exciting. Okay, now we turn to work that's been done in Dr. Anna Johnson's laboratory on behavior. And two kinds of tests. First is the human approach test, also called the HAT test, and the other the novel object test, called the NOT test. And what they do is they will put a novel object, like a traffic cone or a person, into the pen, and that immediate area is called zone 1. And this will be important when you get to the next slide. And as you get progressively further away, so the pig starts out here in zone 4, zone 3 is closer, zone 2 is closer, and then zone 1 is very close to either the human or the novel object. And the question is, how long does it take a pig to go from here to here? Because if the pig is, we'll say, more stressed, then it's likely to take longer to approach this novel object or this human object. And here's what the results look like. Again, some people suggest that pigs that have been selected for superior performance, superior feed intake, are more skittish, they're more high-strung, et cetera, et cetera. Well, you can see here that the duration, the percentage of the time that the pig spent in zone 4, zone 3, zone 2, and zone 1 in the human approach test was the same. There was nothing there to suggest that the low RFI pigs, were any more behaviorally stressed than the pigs that were high RFI pigs. And if we look at the novel object test, again, from zone 4 to 3 to 2 to 1, there was really no difference. Now, some of the other analysis that Anna has done has suggested that there was a slight advantage to the high RFI pigs, but the difference was very, very small. Okay. So let's take a look at meat quality. So this is Drs. Elizabeth and Stephen Lonergan comparing the low RFI and the high RFI pigs. pH 48 hours, there's no difference. the drip loss was higher in the high RFI pigs. The cooking loss tended to be higher in the high RFI pigs. The color score was lower in the low RFI pigs. The marbling score, since they are leaner, we would expect, and the marbling score was a bit lower in the low RFI pigs. And the firmness score, there was no difference between the two lines. Okay. If we look at diet, if you're just interested in what happens when you feed this really low energy, high fiber diet, what did that do? Well, there was no effect on pH. There was an effect on drip loss, and the drip loss was considerably higher in the high energy diet. The cooking loss, however, there was no significant difference, trended in the opposite direction. The color score, there was no difference. The marbling score was slightly higher in the high energy diet, and I think you'd expect that. That doesn't come as a surprise. And the firmness score favored the low energy, higher fiber diet, and we can't explain that. We look at the proximate analysis based online. It really shows us the same kinds of things. The moisture, the difference is very, very small, but there's not very much variation in moisture. So it was significantly higher in the low residual feed intake pigs. The lipid, of course, was lower, which we would have had expected, and there was no difference in the protein content. If we looked at diet, there was more moisture, higher moisture in the low energy, high fiber diet, which is the opposite to what we saw in cooking losses. So I'm puzzled by that one. Lipid tended to be lower on the low energy diet, which we would have expected, and there was no difference in the percent protein. Okay. So in terms of sensory traits, if we take a look at the line differences, there was a slight difference in terms of juiciness, but there were no other significant differences in terms of tenderness, chewiness, pork flavor, off flavors, etc. from the two lines. So we can conclude from this then that pigs heavily selected for improved feed efficiency possess the robustness to be successful in a changing industry, whether we're looking at nutrition, behavior, disease challenge, etc. Those same pigs were slightly inferior in some aspects of meat quality, but while those differences existed, the meats folks were saying that they questioned that would really be detected by the typical consumer. I'm always a little nervous when we say that, because if we keep saying that, you add those on over and over and over again, then you do have something that the consumer is going to pick up on. Pigs heavily selected for improved feed efficiency on a higher energy diet lose much of their superiority, as I mentioned earlier. So let's chat a little bit about the extension component. First off, I hope you're familiar with the book. When we had the conference, I guess 3 years ago now, all of the speakers who participated in that project, Aaron Gaines, who's here, was one of the authored one of the chapters. Heavily referenced, Aaron, by the way, heavily referenced. Actually, what it is, you don't get those kind of numbers, but it is getting a lot. It is getting well read. So every speaker at that conference was invited to. Actually, you were more than invited. You were required to submit a chapter for the book, and this book was published by Wageningen Press, and that book is available, and it covers all aspects of feed efficiency. We also have a website that is all of the research that's being published, all of the publications that are coming out on this project are on this website, very easy, swinefeedefficiency.com. And we're also doing things specifically for the industry. So, for example, there's, I think, 13 fact sheets dealing with various aspects of feed efficiency, and we're working on another 13 right now. And the original 13 have been translated or in the process of being translated into Spanish, because these are really intended, these fact sheets are intended for people that are working in the barn level, so these could be put in the barn. And so we have them translated into Spanish. And then coming up in October next year, not this October, but October next year, we will have our second and final feed efficiency conference. And this will be a conference where we will wrap up the project and talk about what has been found. For example, that'll be a better chance to find out what happened in terms of the genetics component of the project, the results of the new markers or the markers that have been developed, how they're doing when used in a commercial environment. We're just looking for hotels right now, so it'll either be in Omaha or Kansas City. It won't be in Des Moines, because for some reason the conference center, convention center, and hotels don't talk to each other. And we said, we don't want to talk to you separate. We just want to bid on the whole thing, and they don't talk to each other. So it will not be in Des Moines, which is really, I guess, somewhat unfortunate. But I think Omaha did a really, really good job in the first conference. It's going to be a much bigger conference. We will have plenary sessions in the morning, and then we will have up to 4 concurrent sessions in the afternoon, dealing with nutrition, specializing in genetics, physiology, meat science, and so on. But we'll get more of that information out. Hopefully by the end of this October, we'll have the date finalized. We'll start marketing that. And then later this year and early next year, we'll get the program finalized. So that's the project. I just picked one part of it to share with you, but I think it's a topic of interest to the industry. I want to recognize the funding from the USDA and NIFA. And I think it was fantastic. It was quite controversial when they funded this large project, because they funded 3 large projects on feed efficiency, one in swine, one in dairy, and one in beef. And I guess there was one in poultry ultimately too, I think. But anyhow, it was controversial because that meant that there were dollars available for individual projects that individual scientists could be involved in. And so the question is, which is the better use of federal dollars? And I think they're tending to go back to smaller grants spread around more, if my impression is correct. So what I guess I'm saying in part is I don't know if we'll be able to renew this project and continue on at the end of 2016 when it runs out. But I really compliment them, because I think it's just been a fantastic experience for us. I hope you agree that we're able to generate some really unique data because we've got so many different disciplines involved in the project. So thank you very much, Jim, and I'd be happy to try to answer any questions that you might have.
Will skid